In this video, we'll walk through the QIA prep alkaline lysis protocol to extract plasmid DNA from bacterial cells using a commercial mini-prep kit. For this protocol, you will need 2 glass beakers, one labeled bleach and the other labeled kit flow-through waste. You will also need a 1.5 milliliter Eppendorf tube, 2 milliliter Eppendorf tubes, and a spin column with a collection tube. You will need buffer P1, which should be placed directly on ice, along with buffers P2, N3, PE, and EB. Lastly, you will need a 15 milliliter conical tube and your glass culture tube from the last lab period. Vortex the overnight culture on low speed, keeping a finger on the metal cap. Then check if the culture is turbid or cloudy. If it's clear, it likely didn't grow and should not be used. Label your 15 milliliter conical tube, then carefully pour your culture into the tube. Coordinate with another group to minimize waiting time. If you need help operating the centrifuge, ask your IA. Spin the tubes at 6,000 revolutions per minute for 5 minutes in the clinical centrifuge. If you are spinning by yourself, prepare a weighted blank by filling another 15 milliliter conical tube with 10 milliliters of liquid. While your tubes are spinning, prepare a beaker by adding a small amount of bleach. You can also use this time to set up your gel rig and place your elution buffer in the water bath to warm. Retrieve your sample from the centrifuge. You should see a pellet at the bottom of the tube. Carefully pour the supernatant into the beaker with bleach. Use a pipette to remove any remaining liquid without disturbing the pellet. Once the supernatant has sat in the bleach for a short period, pour the bleach down the drain and thoroughly wash your beaker with soap and water. Add 500 microliters of buffer P1 to each cell pellet, using a fresh pipette tip for each sample to avoid cross-contamination. Gently vortex or pipette to fully resuspend the pellet. The suspension should appear consistent when properly mixed. Transfer your resuspended sample into a 2 milliliter tube. Add 500 microliters of buffer P2 and gently invert the tube 10 times to mix. Do not vortex at this step. After inverting, you're ready to move on. The solution should become more clear, but not fully transparent. It's important to continue promptly, as leaving it too long at this stage can risk shearing your plasmid DNA. Add 700 microliters of buffer N3 and gently invert the tube 8 to 10 times. A large white precipitate should form. Then, centrifuge for 10 minutes at maximum speed. Transfer each supernatant into a clean labeled microcentrifuge tube. You will need to then transfer 700 microliters of supernatant to a spin column. You should also have some supernatant remaining. Do not discard this, as you'll run it through the column in 2 rounds. Now, wash the column by adding 750 microliters of PE buffer. Centrifuge for 60 seconds, then discard the flow-through into the waste beaker. After that, spin the column again for 2 minutes at maximum speed to dry it completely. While you're waiting for the spin to finish, prepare a clean 1.5 milliliter Eppendorf tube and label it properly. Transfer the spin column to your newly labeled 1.5 milliliter Eppendorf tube. Retrieve your elution buffer from the water bath and add the recommended volume directly to the center of the column membrane. Let it sit for about 2 minutes to allow full absorption, then centrifuge to elute the plasmid DNA. Placing your tube with the spin column into the microcentrifuge can be a bit tricky. Make sure to turn the cap inward so it doesn't get caught when closing the lid. You'll now have your eluted plasmid sample, ready for the next steps. You can either measure its concentration using the nano drop or hand it to your instructional team for storage.